Welcome to Mr. Chalk's Revision Tips.
When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
Gene mutation is a mutation which is permanent, altering the DNA sequence of a particular gene, such that the sequence is different from that found in most people. Right. It's also worth noting that mutations can be a range of sizes. It's not necessarily just one nucleotide. So when thinking about mutations, so a frame shift mutation, quite importantly, in a gene refers to the insertion or deletion of nucleotide bases in numbers that are not multiples of 3. Right, remember there's 3 nucleotides that go and make up a codon. Right, so this is important because the cell reads the code in groups of 3 bases when it's making protein. So each of these triple codons corresponds to around 20 different amino acids that build up proteins. So if the mutation disrupts this normal reading frame, then the entire gene sequence following the mutation will be incorrectly read. So this can result in the addition of wrong amino acids to the protein or the creation of a codon that stops the protein from growing any longer. So we can see here that we can start off with one particular codon, but if there's a mutation, then it goes and it alters all of the codons after it, meaning that because we've got that frame shift, that all the codons after it go and code for the incorrect amino acids. So a gene mutation can be caused by nucleotide substitution. So the substitution of singular nucleotide can go and change the codon. So whenever we're going into talking about this, always go and refer back to codons. If the new codon codes for different amino acids, this will lead to a change in the primary structure of the protein. So because of the degenerative nature of the genetic code, it might mean, however, that the new codon still can code for the same amino acid. So we can see here that more than one codon codes for the same amino acid. So for instance, if that UUU got changed to a UUC, it would still code for the same amino acid. Second type of mutation is a nucleotide insertion. So an insertion goes and leads to a frame shift mutation. So the triplicate code means that the bases subscribed consecutively will no longer be in the correct order. So this means that the reading frame won't correspond to the correct amino acid. This is also very, very similar when we've got a nucleotide that's being deleted. So if we go and have a look at the question, so a gene mutation is a change of nucleotides within the gene. Well, we've talked about that. Right, so explain, most importantly, how it is possible for a mutation to have no effect on the protein producing that gene. Right, there are a few key things. So we can talk about the fact that some codons or some triplicates code for the same amino acid. So the amino acid sequence will not be altered. It's also worth noting that some alternative amino acids don't necessarily alter the shape of the protein. Right, if we're looking at explaining how a mutation could alter the protein so that it no longer performs its function, we have to go back and think about what we know about protein structure. So, the fact that we're going to get a frame shift in that reading frame, in the triplicates downstream from where that mutation takes place. So the protein will have a different sequence of amino acids, so therefore its tertiary structure or three-dimensional shape will be changed, so it will no longer function in the same way. So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck folks.